I am Isabelle Bonaventure, Analytical Development Manager at Skyepharma. So today I will present you our expertise in analytical development and technical-regulatory and how is it included all along the pharmaceutical development and manufacturing, and how it can help save money and time. So to present ourselves, Skyepharma is your partner for complex solid oral dosage forms. We have a wide range of activities, from early stage development program up to commercial manufacturing and packaging. And whatever the steps, the technical teams, and especially analytical development and technical-regulatory, are involved right at the beginning of the project, and especially since the quotation. And this is a driver of our agility to be operational as soon as the project begins. So, sorry. Okay. Where do I have it? It's okay. So it is our flexibility, adaptability, and agility that help us to target the right approach that will best fit your needs. So we can get you all throughout an end-to-end approach, typically from development up to release to market, or we can help you with specific demands. And as a partner, technical teams will assist you at all steps. Sorry, I have difficulties. Okay. So, in order for you to understand how we work in partnership, I have selected case studies that I would like to show you. So we will begin with the early stage program. Okay. So typically, an early stage program is divided in 4 steps. So I have presented here the first 3 steps and the key milestone for analytical development and regulatory. So we begin with the kickoff meeting. So this is a teaser of your project, and it is done with the technical teams. So then we have the development of formulation, and it takes around 8 weeks to select a formulation. Then we have the manufacturing of the technical batches, and it takes around 20 weeks to launch the stability program of technical batches. So why are we able to select a formulation within 8 weeks and launch the stability program of technical batches within 20 weeks? This is simply because we work as a team, and all throughout our internal meetings, we understand, define, and anticipate the needs of your project. So I would like now to start with the first case study. Sorry, I forgot the 4th step. So then the 4th step is the manufacturing of the clinical batches. And so to summarize an early stage program at Skyepharma, we are able to deliver clinical batches within 7 months. So but just think what happened during these 7 months. In fact, we anticipate for the next steps, the manufacturing of the commercial batches. And because we are working in partnership, the transfer to routine people is smooth. So now let's hear a first case study. So the objective here was to develop a dissolution method during the development of a delayed release formulation. So basically you have 2 options, the online dissolution or the offline dissolution. Usually online dissolution is a preferred option because it is an automatic process. The solution is sampled and read automatically by the equipment. So in that case, we started with online dissolution, and we demonstrated that one of the excipients was interfering with the active ingredient. So option 1 was stop, and then we set up option 2, offline dissolution by HPLC. So the offline dissolution by HPLC was successful in that case, as we demonstrated that there is no more interference between the excipients and the active ingredient. You can see that this is the API peak, and we don't have no peaks from the excipients. So this case study demonstrates that the partnership we have between analytical development and formulation team help us to target the right approaches to develop a complex dissolution method. So let's see another case study for this stage program. Okay, the objective here was to develop a dissolution method for an extended release formulation with 2 active ingredients. So we had a series of challenges here. First, the use of 2 active ingredients, and we had to cover the 5 dosage strengths within these ranges for the finished product. The online dissolution was not envisioned because both actives have the same UV profile. So we then set up the dissolution offline by HPLC. So regarding the HPLC part, we were able to develop a method that covered the full desired range, which is 10% of the lowest dosage up to 125% of the highest dosage. The method is linear and accurate all throughout the desired range. And it uses only the preparation of one single reference solution. So this case study demonstrates that we were able to develop a very efficient method because we have a strong expertise in analytical validation. And for this case as well, we were able to improve the stability of the reference solution. As you can see here, we observed the degradation of the reference solution after 1 week. And by working on the diluting solvent, we were able to increase the stability up to 1 month. So having only one single reference solution stable up for 1 month is really advantageous because active ingredients are very expensive materials. So this case study demonstrates that our expertise in analytical development and analytical validation help us in time and cost savings for the project. So I have demonstrated our partnership approach during early stage programs. So I would like now to demonstrate this partnership during a tech transfer. So from a regulatory perspective, the regulatory strategy is first defined in the early stages of the tech transfer. So first, there is an assessment period of manufacturing process and analytical methods as an anticipation key to target the right approach. So how do we target the right approach? In fact, we can use a scale image to represent that. So on one extreme, you have the conservative approach, which consists in generating and submitting all the necessary supportive data. But this approach is not often compatible with timelines. On the other extreme, you have the risky approach, which consists in providing protocols and committing to later submission of data. This approach can delay the targeted approval date because the agencies will not have the required data. So the strength of our regulatory team is to put the cursor on the right place on this scale. From an analytical perspective, in the analytical development department, we have put in place a systematic approach during tech transfer. So I have presented here the key milestone for the analytics. So first you have a period of 2 weeks for the technical assessment of the methods and writing protocols. Then we have a period of 6 weeks for the transfer of the analytical methods for finished product and qualification of analytical references. And then, if necessary, we can develop and validate the analytical cleaning methods and implementation of raw material. Okay. Then we have a 2 weeks period for GMP documentation and writing risk assessments in preparation of the manufacturing of the validation batches. But once again here, because we are working in partnership, the transfer to routine people will be smoothed. So how do we do an analytical tech transfer? So we can use again the scale image here. You have 4 types of analytical tech transfer. So on the first, on one end, you have a transfer waiver, which is basically a paper-based transfer, and we have comparative testing. So these approaches are rapid, but risky. They should be used under limited circumstances, and the drawback is that you have limited knowledge of the analytical methods at the receiving unit. And this can cause additional costs and use of materials. On the other extreme, you have the revalidation and the co-validation. So these are anticipated type of approaches. They are very useful when you are doing a tech transfer with multi-facilities. And the advantage is that you have a better knowledge of the methods at the receiving unit. And most of all, it covers the changes in the manufacturing process. So again, anticipation. And so the analytical development team is sufficiently agile to put the cursor on the right position on that scale. So I would like now to show you a case study. So in that case, the objective was to transfer a method of content uniformity. The type of transfer chosen was partial revalidation with accuracy verification and intermediate precision. So we ran the accuracy verification and it failed. We obtained recoveries above the acceptance criteria. But finally, during the investigation, we rapidly found out that the failure was due to different lab practice in the preparation of the reference solution. So then, the accuracy verification was restarted successfully. We got the recoveries within the acceptance criteria. So this case study demonstrates that our expertise in analytical validation and development help us to target the right approach for tech transfer. So let's move now to a second case study. In that case, the objective was to transfer an HPLC method for impurity content in a finished product. So the type of transfer chosen was partial revalidation, and when running the analytical method on the HPLC, we observed non-compliant system suitability. We observed relative standard deviation of around 9%, which is far above the acceptance criteria of 5%. But the potential impact of that is that we could underestimate the impurity content on commercialized batches, and this is not acceptable. So finally, we were able to improve the HPLC operating conditions that allowed us for compliant system suitability, but without modification of the regulatory file. So this case study demonstrates that the partnership we build between the regulatory and the analytical development team help us to be very efficient on the ground. So I have demonstrated throughout case studies the partnership approach we have during early stage program and tech transfer. But this is also true during commercial phases. So let's have a look at the life cycle management. So continuous improvement for routine processes. It could sound weird, but it's true. There's always something to improve. So if we have a look at the life cycle management of a manufacturing process, so first you develop your process, then you validate it, and then you go into routine. But one of the most critical steps here is the monitoring of your routine process by trend analysis. And this can only be successful if you have the right people with the right processes. So basically, when you are monitoring your process and you found out that there is a deviation, you have two options. If the root cause is well identified, then you can need to enter into a new cycle of validation and then routine. If the root cause is not well established, then you have to create a new cycle of development, validation, and then routine. So I would like now to show you a case study where we created a new cycle of validation and then routine. So the situation was that one. We are analyzing an active ingredient by HPLC to determine its assay. And in 2018, we observed 30% of non-compliant system suitability, which impacted the batch results. And we could only analyze one batch at a time. So this was not acceptable. We cannot continue to use this method, although it is registered. So we then initiated a request toward our customer to modify this method. And guess what? The supplier had the exact same problem. He was in trouble as well with the stability study of the active ingredient that was impacting the regulatory file. So in the end, we were able to improve the existing method and we co-validated it successfully with the supplier. So what are the outcomes for us? Is that we helped in reducing the release time of the active ingredient at Skyepharma, and we helped to secure the stability study of the active ingredient for our customer through his supplier. So let's see a second case study for commercial phases. So here the objective was to progressively transfer the packaging from a manufacturing site to Skyepharma. So the regulatory approach chosen here was a variation type 1A, immediate notification, after definition of the supply needs with the customer. So for that type of variation, the validation of back transportation was required, and we had a series of challenges during that step. But in the end, the expertise we have in regulatory department allowed us to readjust very rapidly the regulatory strategy, and we obtained the approval within the expected timelines. So once again, this demonstrates our agility and flexibility. So it is now time for me to conclude. So from all the case studies I have shown you, I would like that you remember that because we are working in partnership, the analytical development and the regulatory team are a warrantor for routine processes. And how do we do that? Thanks to the 3P. We have the right people with strong expertise to target the right approaches. We have the right processes to be efficient. And finally, we build a strong partnership internally and with our customer. So I invite you to come and meet us at our team on our booth, 120 F75, and thank you for your attention.